the signals from the sensory neuron are sufficient to produce the appropriate reflex behavior and change in contraction of the muscles. So you should be able to find sensation, conscious production of movement, reflexes. Connections between the cells in these networks, the flow of signals through those networks is what produces our behavior and our thoughts and conscious experiences. Now, by looking at pictures like this and all those drawings I made of networks, you might get the picture in your mind that these networks are static structures. Synapses are made. They're well-worn paths through which signals go. That is not correct. Like all parts of biological system, these networks are dynamic. They move. They turn over. These axons and dendrites are continuously extending, collapsing, retracting all the time. Reaching out, pulling back, reaching out, pulling back. Everything is moving around. You are continuously getting new synapses forming. You are continuously getting old synapses breaking, disappearing. Now, what happens when you have a dynamic system? Any dynamic system. What property comes out of these dynamics? Plasticity. We saw it in the skeletal system. We saw it in the muscle system. And the nervous system is the prime example of plasticity because things are always changing and dynamic. The nervous system can be molded, can change its structure, can change its function, depending upon what's going on around you. So what emerges from this is plasticity, and particularly brain plasticity. And behaviorally, you should automatically know that the brain must be plastic because you can learn. You can know something that you didn't know before. When you leave this room today, you will leave knowing something you didn't know when you walked in. You acquired knowledge. You acquired new thoughts. You acquired also, hopefully, new memories. If all behavior and all memories and everything going on in your head is the flow of the signals through some network, where were those networks before you came into the class? Where were they? Nowhere. Right. They didn't exist. But when you leave this class, all these new thoughts and ideas and things you've learned and memories you've formed, that's the result of signals flowing through new networks that were not there before. Plasticity. I molded your brain today. Hopefully, those of you who fell asleep did not get molded very well. Well, it looks like most of you are awake. Okay? plasticity is a most important aspect of nervous system function in our behavior. We have to have dynamic networks. Now let's look at how this plasticity arises. What happens in the brain when a new network forms that enables new networks to form? So for example, one way we see neuronal plasticity at the cellular level, we see this in terms of the branching in different neurons, changing depending upon how they're stimulated. So if this neuron is probably not being stimulated a lot, this is a neuron in the cortex, part of certain networks that are not firing a lot. This neuron is part of a network that is firing a lot, is being used a lot. It has many more branches. And if you look at those branches at a higher power in a microscope, you see these spines, dendritic spines. What those correspond to are postsynaptic contacts. There is an axon that's not being shown in this picture, making a contact with each of those postsynaptic dendrites at those spines. So if this branch has many, many more spines than that branch, it's because it's making more synapses. It's been stimulated. These spines and these synapses have been stabilized. So in the course of this class, this session today, hopefully many of your neurons are having more spines attached to them, more synapses connected to them. In addition, depending upon how it's stimulated, the amount of stimulation, you can affect the size of the synapse. A larger synapse that has been stimulated a lot will conduct signals through the network a lot better than a smaller synapse. So plasticity is expressed in the size of the synapse, the ease with which signals flow through a network involving larger synapses. In this picture you'll have to look at carefully. You all have a copy of it. This actually shows the changes in a network which occur in experimental animals that are trained to perform a certain motor task, learning how to use their hands in a different way. Here is the state before any training. There, here, two different situations, two different examples of this. Look carefully at the postsynaptic dendritic spines, that's in blue, and the other branches are the axons. Look at the synapses. Look at the points of contact before training. Now, the animal is trained and has learned how to perform a new task. Whoa! Here's a synapse that didn't exist before. A new synapse formed. Here's a synapse that disappeared. Here's another synapse that might have existed before, but certainly got much bigger. This is a very different network from the one that existed before training. And then you might see, well, that's just because the animal is doing something, anything. This might not be related to a particular task. So what you do is you teach the animal a different motor task, a different way to do something with its hand. And you teach it the second task, and you look and see what kind of changes occur. And if you look carefully, you can see right here, a new synapse is formed, which was not present after learning the first task. Another synapse, which was present before, disappears. All this making and breaking of synapses is an expression of brain plasticity, carried to its extreme in the brain, to which we take the most advantage. humans smarter than other animals? Are our brains more plastic than other animals? Some might think so. Some might think our brains are too plastic and that we're too wishy-washy and change our circuits all the time. Certainly in times perhaps when our circuits should stay the same. It's an interesting question to debate and argue. Okay, so now we've covered our discussion of these networks and their plasticity. And we said mental processes, ideas, and everything is the result of signals flowing through these networks. What are these signals? Now this will probably be the lecture that's the heaviest in terms of real physiology that we're going to be covering. And every time you find yourself getting bored or your attention lagging, I want you to go back to the big, hard problem, the question. Remember the question. That is, how do signals flowing through networks relate to a thought in my head? They're two different things, it appears, but yet one is equivalent to the other. And now as we go back and start explaining what these signals are, just think of them in terms of, well, at one level, this is me. This is my personality. This is my memories. This is my thoughts. And if you keep that in perspective, you won't get bored. Promise. Okay, so now we want to get signals flowing through networks. How do neurons do that? There are certain properties that neurons have that other cells do not have. Things that make a neuron a neuron. We call them neuron-specific functions. There are 4 of these neuron-specific functions. And they are what enable neurons to form networks and to have signals flowing through these networks. First one, axonal conduction, is the ability to conduct or carry signals over long distances. The longest part of the neuron is the axon. The axon has the ability to conduct signals along this great length from the top of my head to the base of my spinal cord, from the tip of my toe to the base of my spinal cord. Those are some of the longest axons, and they can conduct signals those great distances. So here is a network. That's axonal conduction. Conducting signals that great distance. But neurons have to do something else to get signals flowing through a network. They have to get the signal past the synapse. Synaptic transmission. Synaptic transmission is sending signals from one neuron to another. From the axon of the presynaptic cell at the synapse to the dendrite of the postsynaptic cell. That is synaptic transmission. So you get the axonal conduction, getting the signal long distances to the end of the axon, but then you have the second, and as we'll see later, totally different process that gets the signal from one cell to the other. Then, the next thing that cells have to do is called neuronal integration. This is the ability of each neuron to analyze all of its inputs and to decide whether to pass a signal on. So, in reality, each neuron receives synapses from many other neurons. And realistically, sometimes thousands of neurons will synapse on any given neuron. Some of those synapses will be excitatory, some will be inhibitory. Each neuron in its dendrites and in its cell body must take all these inputs, thousands of inputs, analyze them, and decide, should I pass the signal on, or should the signal stop here? There's not enough excitation. Okay? The fourth thing that has to be done is specific synapse formation. These cells, the neurons that are going to wire themselves up into networks, have to be able to recognize and distinguish one neuron from another. They have to be able to carry out cell recognition and decide, should I make a synapse with this cell or shouldn't I? And this is very important, because if cells do not wire up correctly and form the right networks, the whole nervous system is miswired. Inappropriate behaviors might occur. You might say, oh, I've got an itch behind my ear and want to scratch the itch, and every time you go to scratch the itch, your foot kicks. If the neurons are not connected up correctly, my ear is still itching. It's not working. And that thing's not going to wire up. Or, instead of hearing my voice, wouldn't it be amazing, instead of hearing my voice, you saw colors, didn't hear words? What if the sensory neurons that bring sound signals to your auditory cortex, which are then carried to Wernicke's region, what if they went to another part of the brain? What if they go to the visual cortex instead and activate those neurons? Then you're going to see me in Technicolor. You're going to hear me. You'll see what I'm saying. You'll see what I'm saying. Yeah, it'll be very strange. You've got to get the networks wired up correctly. And let me just tell you how that's done, because that's something that takes place during development. Remember we talked about molecules, proteins that have lock and key interactions? Well, on the surface of all cells, there are molecules. One cell that has a certain binding site, and on this cell, there might be a molecule that has that shape. These two molecules on the surface of the cell fit together like a lock and key and adhere to each other. This cell, let me give you another one. This cell might have on its surface molecules shaped like that. This cell will not be able to recognize and form a synapse with this cell because the molecules on their surface don't interact in that lock and key manner. So it's interactions between these molecules on the surfaces of the cell that enables the correct cells to wire up with each other. Okay, so 4 functions that neurons must be able to perform to form these networks and have signals flowing through them. So to have a signal go through a network, there are actually at least 3 different paths. 1 along the axon, 2 at the synapse, 3 along the dendrites in the cell bodies. Axonal conduction, synaptic transmission, neuronal integration. 3 different paths, 3 different kinds of signaling. And I want to go over and tell you now what produces the signals along each of those different paths. And we'll start off with one, along the axon. Now, the way we discovered how signals travel along axons derives from an accidental discovery. An accidental discovery that was done in the 1780s, a long time ago, by a professor of anatomy named Galvani. And what Galvani was studying in his lab was the structure of muscles in the leg of a frog and the structure of the nerves that presumably carry motor signals to those muscles in the frog's leg. Okay? So he had a lot of people in his lab, partially dissected frogs. You didn't do that in your labs, did you? Too bad. A lot of fun. Pull apart a frog and look at its nerves and its muscles and so forth. Now, Galvani, in addition to being a professor of anatomy, and this is not a contradiction in terms, he was a cool guy. He liked to have a good time. And in the 1780s, one of the cool things to do, what the cool people did at parties, was to play around with static electricity. You all know how if you take a comb and rub it against a wool sweater, it'll be able to pick up small pieces of paper or make a little spark. If you have the lights out, you can see it. Well, at this time, they developed these very elaborate machines for generating static electricity. Here is a wheel. Instead of just rubbing something against a sweater, they had a wheel attached to a handle that they could crank and get this wheel going really fast and rubbing against something up here and generating huge amounts of static electricity, which would build up, and then they could send through wires to anywhere in the room and zap people with the built-up static electricity, and they would jump and everybody would laugh, and this was a great party. You think you guys have fun at your parties on weekends. They knew how to have a good time in the 1780s. Now, as I said, Galvani was a cool guy, and he liked to mix his work with his pleasure. His work was doing the structure of frog muscles and nerves, and the pleasure, he got his kicks by zapping people with static electricity. And he'd have graduate students working in his lab, and if they weren't working well, or even if they were working well, he'd like to zap them anyway. Well, one day, he did this, and he missed. He didn't zap and hit the graduate student, but instead, he zapped and he hit a metal surgical instrument that the graduate student was holding in his hand, and that metal instrument was attached, touching, almost like here, a nerve. And when electricity hit that instrument and that was attached to the nerve, all of a sudden, whoa, the frog started moving. Muscles started contracting like it was alive again. This is probably the basis behind the Frankenstein story, which probably wasn't written until the 19th century, almost 200 years. Yeah, 100 years later. The idea of, bringing the dead to life again by shocking them with electricity. Accidental observation. Electricity hitting a nerve could make muscles contract. Galvani, very smart guy. He, from these observations, had led him to the hypothesis that animals, not only can they be stimulated by an artificial external source of electricity, but he took it a step further. He said that animals can produce their own electricity. That this is how the nervous system works. This is how the nerves send signals to the muscles and make them contract. He said that some of this animal electricity this is equal to the nerve signal. The nerve signal that passed on to the muscle causes the muscle to contract. What an idea! Prior to this, nobody had any idea. Now, even probably at this time and even right now, people don't have a good idea of what is electricity. What is this thing that travels through a nerve and produces contraction of muscles? Well, we're going to use a very, very simple definition of electricity. Electricity, we are going to define as the movement of any charged particle. And if you don't understand what a charged particle is, don't worry, because I don't know either. It's hard for me to explain to you what a charged particle is, other than we know about electrons, we know about ions. They have an electrical charge, not an equal number of positive and negative charges. They move when they're exposed to a battery, to a force, but that doesn't really tell you what a charge is. Okay? It's just a concept that has been generated. So, there's movements of some kind of charged particles along the nerves. How are we going to measure them? How are we going to see them to make sure that this is what's going on? Well, there's two things you can measure. We can ask, how do we measure electricity? One thing you can measure is the number of these charged particles that are moving. And this is what we mean by current. Electrical current is how many of these charged particles are moving. The other thing that we can measure is the force that pushes the particles. Nothing is going to move. No charged particles are going to move and make electricity unless there is something pushing them or pulling them. There has to be some force acting on these charged particles. This is what we call voltage. And there are instruments. We have developed instruments that can measure current, or they can measure voltage. And what we want to do is to take these instruments and stick them into a single cell and measure the electrical properties of a single cell, of a single axon. Now, that's amazing. And we can do it. And if you go on to a neuro track in biology here, we have lab courses where you actually get to do that. And what we do is you take a small glass tube. Small glass tube. Remember, these cells are microscopic. You can't see them with the naked eye. This glass tube is not microscopic. You can see it with the naked eye. But you heat it up over a flame, and then you pull it out. And it melts, and it gets thinner and thinner and thinner until it's so thin you can't see it with the eye. You have to use a microscope to see it. And it's so thin that you can stick it through the plasma membrane of a cell without really damaging that cell. Here's an example of that. Here's a microelectrode penetrating this cell in the cerebral cortex. You can't, by eye, even see the tip of that electrode actually going into that cell because that electrode is even smaller. That's going to go right through the plasma membrane, and it's going to measure either current or voltage. We call those kinds of electrodes